Welcome to all for this International Symposium on Advances in Computational Neuroscience. Today I'll be talking about some work we've been doing on modeling the basal ganglia to understand Parkinson's disease. So I come from Department of Biotechnology in IIT Madras. So my lab is called Computational Neuroscience Lab. Our long-term goal of my lab is to build a simplified whole brain model. Once it is developed, we believe that it will have immense applications in both medicine and engineering. So the overall architecture of this model will be something like this. So there are these sensory ascending sensory streams on one side, the vision, touch, and sound. Other side, you have the descending motor streams, musculoskeletal system, speech, eye movements, and all that. Between the two, you have this huge decision-making regions of the prefrontal cortex. And if you can think of the whole thing as a massive feed-forward pathway. And then there are three loops, the cerebellum, basal ganglia, and hippocampus. You can see that it's a hugely simplified depiction of the brain, but this is like a prototype of the full brain, right? So if you can have this and have represent each of these structures, and first of all, we need to develop the corresponding theory for each of the structures. Because you ask anyone who has worked on any of the brain structures, for example, if you ask Dr. Shyam, he will tell you how complicated cerebellum is and how many challenges are still left. So I'm talking about overcoming those challenges and coming up with a kind of an essential model, a basic model of each of these brain structures, assemble the whole brain model. And once you do that, from such a model, you should derive various disease models, because in disease, some part of the brain gets affected, and so you need to damage it appropriately. And then you should be able to link such a disease model to the corresponding symptoms. That will give you an insight into what exactly is happening in the brain, the corresponding disease. With the help of such a model, we hope to develop better therapeutics, more logical and quantitative therapeutics, rather than the empirical and often hit and trial kind of therapeutics that you see for most brain disorders. To this end, my lab is divided into multiple teams. Each team works on one problem with a specific goal. So one of the teams works on basal ganglia Parkinson's disease. So that's what I'm going to talk about today. So basal ganglia is important because it is involved in, so although it's a very small subcortical circuit, it has widespread contributions to various brain functions. In fact, it is involved in all the four major domains of brain function. That is sensory motor function, cognitive function, affective or emotional function, and autonomic function. That is control of the internal organs. So it's quite challenging, quite fascinating to know that such a small brain system can have influence on pretty much everything that brain does. So it's quite challenging to understand how it is possible. So our goal is to understand Parkinson's, understand basal ganglia in all its facets, and then with that understanding, with such a quantitative model, develop treatments for Parkinson's. So like I just said, basal ganglia is a subcortical circuit. It takes inputs from the cortex and returns in outputs back to the cortex via thalamus. And the inputs coming from the cortex travel through two different pathways within basal ganglia. So input port of the basal ganglia is called the striatum, which has again two components, putamen and gross and caudate nucleus. And for the output, there are two output ports, GPI and SNR. The outputs of the GPI and SNR then project to thalamus, which in turn projects to cortex. So now there is a direct pathway which connects striatum to GPI or SNR. So therefore, it's called the direct pathway. Then there is also a longer route which connects input port to output port. This goes from striatum to GPE to STN to GPI. So this longer route is called the indirect pathway for obvious reasons. Then there is a very tiny cluster of cells called the SNC. These cells release a chemical called dopamine, and these cells project to pretty much all targets in the basal ganglia. So in Parkinson's, there is loss of some kind of inexplicable loss of cells in this nucleus, the SNC, which seems to give rise to all these symptoms. So like I just said, the functions of basal ganglia are multitudinous, and in fact, this is what prompted British neurologist Marsden to comment about the mysterious motor functions of the basal ganglia. So it's not just motor functions, it has functions in so many other things. So it's quite mysterious because how can it have such widespread influence? So there have been computational models proposed, but very often the models target into individual functions without any awareness or consideration of how the same model can explain the functions of basal ganglia in other domains. Suppose you have a model of reaching, the basal ganglia's contribution to reaching. How do you explain its function, its role in working memory or decision making? So these are the kinds of questions, right, which is generally not answered because models, as often they are quite exclusive and are addressing only one function. So if you look at classical literature, because modern literature in basal ganglia goes back to at least the 80s. So if you look at a lot of the early papers in the first few decades, it's you see it's like this, monks figuring out the shape of an elephant, right? Somebody thinks it's like a pillar because they're only looking at the legs. Somebody thinks it looks like a rope or a snake because they're only looking at the trunk and so on. So what is needed is an integrative picture which can reconcile all these different facets of basal ganglia and give a complete picture, right? Now this is why it behaves like this or functions like this in various domains. Now coming to PD, it's a progressive neurodegenerative disorder because it happens because of loss of cells. We don't know why these cells die, therefore it is called idiopathic, because idiopathic simply means I don't know what's happening. So there are these cardinal symptoms, the key symptoms of Parkinson's, right? This is tremor, rigidity, postural abnormalities, and bradykinesia. So and within the motor symptoms alone, because I said the symptoms show up in all the four major domains, but within the motor alone, you can have problems of reaching, which again, more specifically, it can show up in handwriting movements and eye movements. So they can have nystagmus and speech. They have some kind of dysarthria, then posture, then gait. Right. So it can have many symptoms within many. So many of the motor modalities themselves can be affected. So on the right, you see handwriting samples from PD patients. So above, this is a Tamil word Sri Ramajayam. Above it is written by an elderly, healthy control, a healthy gentleman. And below it is written by PD patients who are age matched. You see that the letters are quite small. It is called micrography. So let us begin with the classical view of basal ganglia. Then I'll talk about how we've been trying to kind of modify that and expand that classical picture. And I'll also show how our expanded picture can explain wide variety of functions and can reconcile these apparently unconnected or irreconcilable functions of basal ganglia. So like I said, basal ganglia has anatomically, there are two functional pathways, the direct pathway and indirect pathway. Direct pathway going from striatum to GPI is straight, and indirect pathway going from striatum, GPE, STN, and GPI. Now you see that the projections of SNC, the dopaminergic projections from SNC go to striatum. Now there's a very interesting cellular machinery within striatum, and when dopamine is projected to striatum, it acts like a switch within striatum, or you can say a valve, which will enable the information coming from the cortex, right, the arrow coming from the bottom, to go either straight through the direct pathway or go through the longer route, the indirect pathway. And I'm simplifying it a little bit, but that's essentially what happens. So therefore, if there is high dopamine, the information goes through this direct pathway. This low dopamine information goes through the longer pathway, the indirect pathway. So now when the direct pathway is selected, right, let us see what happens. So there is an inhibitory pathway from striatum to GPI, which is a GABA projection. From GPA onwards to thalamus, again, it's GABA, right? So you have two negatives, which makes a positive. So when the indirect pathway is selected under conditions of high dopamine, the movement gateways are open. So the movement gets facilitated. You are able to move. On the other hand, if you look at the indirect pathway, you have GABA here from striatum to GPE. Again, from GPE to STN, it is GABA. And from STN to GPI, it is glutamate, which is positive. Again, from GPI to thalamus, it is GABA. So you have three negatives. So on the whole, it becomes a negative. So so it is on the whole an inhibitory pathway. Therefore, when the dopamine is low, that pathway is selected, and therefore movement is inhibited. Right. So therefore for these reasons, they call the direct pathway the go pathway and the indirect pathway the no go pathway. So this is a classical picture. I can tell you straight away that it is oversimplified, and the function of basal ganglia cannot be reduced to simple binary terminology where the direct pathway is enabling movement and indirect pathway is inhibiting. It's not as simple as that. To understand this broader picture of basal ganglia, we need to first understand the significance of dopamine. Right? What is it and what does it do? What does it mean? What is a dopamine as a chemical signal? What does it mean? So dopamine is called a pleasure chemical, brain's pleasure chemical. So whenever you feel pleasure, there is a dopamine signaling, dopamine release going on somewhere in the brain. So, for example, it is known that dopamine release is triggered by brain stimulation. Here by brain stimulation, what we mean is there are certain centers in the brain which have been known for several decades, which, if you electrically activate, it releases chemical, dopamine, and that will make the organism feel kind of happy. So, how do you know that it is happy? So, for example, in some experiments, so the animal is hooked with this electrode, which is inserted in one of these so-called pleasure centers of the brain. And the animal can press a lever and which will activate this electrode, and there's some current that is passed into the center. So the animal repeatedly presses the button and keeps activating itself, and it seems to be satisfied by that. It will keep on doing that even foregoing food until it literally starve itself to death, but it is happy just by pressing the button. It's almost like a drug addict. It wants only one thing, that stimulation of its pleasure centers. All right, so so that happens because of dopamine release. So it's a part of this something called the reward system. Similarly, psychomotor stimulants, opiates, food, even food is pleasurable. Very often we eat even though we are not hungry because food is pleasurable. And all these things that cause pleasure, sometimes even pathologically, activate the reward system, and dopamine signal is a key player in this reward system. So contrarily, if you apply dopamine antagonists, that is chemicals which block the action of dopamine, that will attenuate the pleasurable experience of the above. Right. So, so it's clear the role of dopamine in pleasure is quite clear. In addition to these kinds of primary stimuli, which can give you pleasure, even stimuli, more higher level stimuli, cognitive stimuli, like presentation of beautiful faces or images of lovers. If you take a couple, obviously each of the couples, they find the other person most attractive and so on. So when you see beautiful faces, and people have done this in functional imaging, if you scan the brain, you will see that the same pleasure centers or reward systems are activated under this stimuli. Similarly, monetary rewards. People crave for money because money gives us kind of a high. The reward centers are activated. So in all these things, dopamine plays a key role. So this aspect of dopamine is known for quite some time, but there is a seminal experiments by Schulz et al. Have revealed something even more subtle about the meaning of dopamine signal. So in this, it was done with monkeys. So the electrodes were placed inside a dopamine center called the ventral tegmental area, not SNC, right? So this is called VTA, and so in the beginning they kind of give a flash of light, which is called a conditioning stimulant. The flash of light in itself is not reward. So if you just give it a flash of light, that doesn't cause activation of dopamine cells. But the flash of light is paid by a reward signal, which is actually presentation of a piece of apple to the animal, right? So what happens is in the early experiments, they give a flash of light, and after some time, piece of apple is given. When the moment the apple is given, the dopamine cells fire because the apple is food, and that means reward. So the cells fire in response to that. But after some initial trials, where this both are presented like this, conditioned stimulus and the reward stimulus, so they see because of this pairing between the conditioned stimulus and reward stimulus. In the early trials, these neurons fire only when the reward stimulus is given. But after some trials, you see that the time of firing of the dopamine cells, it migrates and gets and advances to the time of presentation of the conditioning stimulus. So that means the moment you present this flash of light, the neurons start getting activated. So you see the high firing rate here, which means that the neurons are anticipating that the reward is given. So the neurons, with this firing of these neurons, it doesn't just represent the arrival of the reward, but only it can even represent anticipation of reward. So it knows that reward is going to come, so it's already expecting that and firing. So that's a slight extension beyond our basic understanding that these neurons firing represents the coming of the reward. Okay, so in the third set of trials, they've done something interesting. So they did the same experiment, but at the time of reward presentation. So that is, the animal actually has to put its hand in a box and pick up the piece of apple. So in third trials, they actually cheated the animal. They kept the box empty. There's no apple. So the flash of light is given. The dopamine cells are firing. The animals are expecting reward. When they actually put their hand in the box to pick up that reward, they found that the box is empty. So they are disappointed in some sense, right? So when that happens, the firing rate actually goes down a little. So which means that the dopamine cells is they're not the firing doesn't just represent the coming of reward, arrival of reward. It's not just the anticipation of reward. It seems to be a difference between the actual future reward and the expected future reward. So in the last case, the future reward is low, right? The expected reward is high. So there's a difference, and there's a negative value. So there's a dip in the firing activity. So this indicated that dopamine cells firing represents a reward prediction error, the error in reward prediction, and not just the reward itself. So moment you have this kind of a, association between the dopamine cell firing, which is a physical thing, and a conceptual, a mathematical quantity like reward prediction error, right? You could link it to a huge body of machine learning called reinforcement learning. And so that's what people have done. They applied ideas from reinforcement learning and developed the theories of basal ganglia. There's a huge amount of literature on those lines. So what is this reinforcement learning? So let us give again, because it is inspired by animal psychology, the early studies on animals and how you can train animals to learn something. All right. So how does the experiments work? So animal is presented some stimulus. In response, it produces some response. And because of the response, it gets, depending upon what the response is, it gets either reward or punishment. So suppose the response gives it reward, so that green arrow here. So animal understands that for that stimulus, this response is appropriate. So that will reinforce that connection. So next time around, it will repeat the same response. On the other hand, if the response results in punishment, the animal understands that this is not a good response. Next time I try not to do it. So the stimulus response connection will be attenuated. So this, simply put, is reinforcement learning. But as you elaborate this idea, you get very sophisticated body of mathematics, and which, when you apply to basal ganglia, it gives a lot of insights into what basal ganglia does. Now we can straight away see, without even looking at having any detailed understanding of basal ganglia, its anatomy, its neurochemistry, and all that. Just by a glimpse of its anatomical position in the brain, you can already see that it is potentially a reinforcement learning machine. Why is that? Because if you see where it is placed in the anatomy, it gets input from the sensory motor cortex, right? So sensory cortex tells you what is the stimulus. Motor cortex tells you what is the response or what have you just done in response to that stimulus. Then the signals from essence, a dopamine signal, which carry reward to basal ganglia, they tell you, okay, what you have done, is it a good thing or a bad thing, right? So all these three signals meet in basal ganglia, just like all these three things meet in this box. So you can easily kind of guess that basal ganglia could be indeed involved in reinforcement learning. Let us develop these ideas a little bit more. So imagine a monkey faced with two choices, right? Pressing a yellow button or a red button. If it presses yellow button, it gets reward. Red button gives its punishment. It doesn't know that initially. So initially it thinks that both are equally good because it has some kind of a priors on each of these choices. So it presses the yellow button, it gets some orange juice. So it understands that yellow is good. So the weightage for yellow goes up. Then it presses the red button, it gets a shock, which is punishment. So it understands that red is bad, so it goes down. So yellow is like go or reward, and red is like no go, punishment, right? But if it did only these two things, that's not going to be a comprehensive system or a strategy for action selection, for making choices. It needs to have one more capability, which is explore random choices to see when any of those choices also lead to good rewards. So let me give an analogy from our day-to-day life. So suppose we all go to restaurants, right? We all go to restaurants to have a good experience, have good food, good ambience, and all that. And we know that when you go to some restaurants, you end up sick, you fall sick, and because there's something bad about the food. And so some restaurants give you a happy experience. It is, you get the bang for the buck and so on and so forth. So based on your past experience, you mark some restaurants as good restaurants. You tend to repeatedly go to those restaurants. You mark some restaurants as bad restaurants, try to avoid those restaurants. But if you stop with that based on your past experience of restaurants and only keep on visiting those restaurants or avoiding those restaurants, then doesn't maximize your restaurant experience, right? The rewarding experience of restaurant, because you should always be willing to try out new restaurants, because there could be new restaurants which are constantly keep coming up in your city, which are better than some of the best that you have seen so far, right? This willingness to try out new choices, which might give you even greater reward than what you have got from the positive choices from your past experience. This capability is called exploration or exploratory ability, right? So an animal or an organism or even artificial agent, which is trying to maximize its reward by making choices, should be also designed or should be willing to try out new choices to explore the world and constantly seek more rewarding experiences. And with this, we know from day-to-day experiences. But these ideas are quite important in the understanding of a reinforcement learning system or an agent. So what we need for a reinforcement learning system is there must be a reward signal. In case of basal ganglia, that's the SNC dopamine signal. These weightages the animal is learning. So it understands that yellow has high weightage and red has low weightage. These weightages are called values in reinforcement learning, for want of a better term. It's a very simple term. Some can be confused with, right, because it's a very general connotation, so it can even be sometimes confusing. Then the ability to explore various actions, unknown and try out unknown choices. And action, you need to have some way of making an action, and that comes from motor cortex, and some representation of the state, that is information coming from the sensory state. So where are all these things sitting inside basal ganglia? Right, basal ganglia is a complete reinforcement learning system. So that's the premise with which we started. All right, where are all these things sitting in the basal ganglia? So now sensory information, the state, right, comes from the sensory cortex. The action is ultimately the GPA output has to go to thalamus to motor cortex, and that's where you make an action actually. So you can say that somewhere there is an action representation sitting there. Then so where are the values? So we learn something, these weightages that the animal learns, where are the setting? So I don't have those slides, but neuroimaging studies on subjects who are playing some kind of a gambling game, like it's a standard task called Iowa gambling task. Right, there also you have some kind of value representation. You apply a reinforcement learning model for the subjects playing the game. Right, people have found, Doherty and Nathaniel Daw and others have found that the striatum or ventral striatum part of basal ganglia, the activity there seems to correlate very well with what we call value in reinforcement. So that's a now a well accepted thing that the striatum encodes these values. Now dopamine signal is here. So what is missing is exploration. Where is exploration happening? How is exploration being implemented? How is it driven? So exploration means some random, making the ability to make random choices. You have a bunch of choices, I would randomly pick something. In a computation model, you just use as a kind of a stochastic model for action selection. That's a mathematical model, but biologically, how is it implemented? This was the question we were facing when we got into the field, right? So because the classical picture is this, the go pathway facilitates movement, and a no-go pathway blocks movement, and that's all there is to it. But like I said, this often this simplistic binary understanding is justified in terms of the neurotransmitter stages. Like I said, you have two negatives on the direct pathway side, so it's on the whole positive. Three negatives on the indirect pathway side, so on the whole it is negative, and so on and so forth. So they try to justify it. But this picture is not quite, the arguments are not quite complete, because on the indirect pathway side, you don't have just a serial order, right, of projections. There's also feedback from STN to GP. This feedback brings some complications or also some interesting opportunities, because of the feedback, the SNGP forms a loop, right? And this loop is capable of producing oscillation, because SNGP forms a loop of excitatory nucleus and inhibitory nucleus. And a loop of such a pair can produce oscillations, which is known both in mathematical literature and in biology, in neurobiology also. So with that, we didn't conclude straight away from that. We did a lot of work before we came to this conclusion, but I'm just giving a basic idea here. The idea is that the indirect pathway is not only, I think we should call it not only the no-go pathway, but also the explore pathway. Because this is the one, by virtue of its complex oscillations, is able to drive exploratory activity in the output. Right. So let us see. So we proposed quite a long time ago that the basal ganglia function must be, you shouldn't just talk in terms of go and no go, but should talk in terms of go, explore, no go, or gen. So we call this the gen model of basal ganglia. So how do we say that the SNGP is involved in exploration? So let us look at some preliminary evidence right before we can go deeper into this. So it is known from studies of Griller et al., Stefan Griller and others, that decorticated ketones show exploration. There's a special preparation called decortication. Right, so you kind of cut off cortex from the output. So whatever output the animal is producing, it is not coming from the cortex. So even such a ketone is able to explore and hunt for food. So which means exploration is not just cortical. Then STN lesions cause what is called perseverative behavior. So perseverative behavior means that you're keeping on doing the same thing even though it is not giving you a reward. Right? In fact, there's an interesting quotation from Einstein. I don't have the quote, but he defines this as insanity. Insanity means doing the same thing over and over again, although you know that it doesn't work or it giving you a negative results. So in perseveration, what happens is, in a psychology or a psychiatry context, right, the patient keeps on doing something even though it doesn't give reward, right? It keeps on persevering in doing that, although it's not working. Then in terms of electrophysiology, so people have shown that if you measure the activity of SCN and GP, the activity under dopamine deficient conditions, which is what you see in Parkinson's disease, there's not much change in firing rates, but there's a increased correlation among the activities of both STN and GP independently. There's higher synchrony levels within STN. In fact, in PD, you find synchronized oscillations in STN in the range of beta, that's around 20 hertz. You will see some of those results later on in the talk. So in modeling also, people have explored this aspect or studied this aspect of SNGP extensively. Some of the early models, they've looked at this model SNGP as a pair of accelerator inhibitory, nuclei, and have shown that such a system can produce oscillations. And others like Terman and others have shown using conductance-based models of SCN and GP, they have shown that by varying some parameters of the network, the system can be shown to produce complex spiking patterns, traveling waves, and synchronized bursts. So with this background, I'll just present a simple model of basal ganglia, and with which you can, so whatever kind of hypothesis I've just described, right, that the basal ganglia function must be described in terms of go, explore, and no go. You can see those effects coming out of a very simple model of basal. So in this model, there is striatum, and so there is a GP, STN, and the output says GPI goes to thalamus, and then you have final output. And dopamine signal is coming from SNC. So what are we giving to this system? We're giving it two choices in the striatum. Imagine these two choices are actually values. It's already computed values, let's assume. And you have given two choices. One has high value, that is choice A. B has low value. The system should choose A, right? That's what you expect it to do. Now I vary dopamine as an independent parameter and say what is the effect of varying dopamine on the action selection. So let's look at some of the modeling features. So dopamine projects to striatum, and striatum has interestingly two different kinds of cells which have dopamine receptors. So these cells are called projection neurons of striatum. They project outwards to GPI and GP and even SNC. There are two kinds of these cells. These are called medium spiny neurons. There are two kinds of medium spiny neurons or MSNs. There's one class which has D1 type of receptors, another class which has to D2 type of receptors. The D1 type of receptors basically increase the firing rate when there is dopamine. So when the input is the same, when there is high dopamine, the D1 MSNs will show even higher response. Whereas the D2 MSN show the opposite effect for the same input, right? When there is an increased dopamine, the D2 MSN show a lesser output. So we model this by simply modulating what is called the transfer function of this. So we just model this as rate-coded neurons, and we set dopamine to simply control the slope of the transfer function of these two cells. Then STNGP, we model this as a pair of inhibitory and excitatory systems. And we show that by varying this cross connection strengths, you can produce oscillations in STNGP. If I take only one pair of units, one STN, one GP, you get limit cycle oscillations. But when you couple a network of them, you get a lot of rich behaviors like you can get synchronized oscillations, you can get chaos, you can get traveling waves, and so on and so forth. So we have characterized the system separately in separate papers before, right? And also by increasing input from the striatum, you can produce oscillations. By increasing the input from the cortex to directly to STN, it is called a hyperdiric pathway. Again, you can trigger oscillations and so on. All the things that we know, that some of the basic effects that we know about oscillations in STNGP, we are able to reproduce in this very simple model of STNGP. Then you have output, which is here, like in GPI. The outputs of both direct pathway and indirect pathway merge in GPA, then that goes to the thalamus. Okay, so when we did this simulation, right, what we observed is when the dopamine levels are high, so we wanted to choose A and not B, right? And that's what the system does pretty much mostly when the dopamine levels are high. When dopamine is like, let's say dopamine varies from 1 to -1. It's an abstract number. At 1, it almost always chooses A, right? But as you decrease dopamine levels, something interesting happens. And there are many times where instead of A, it chooses B, which is not the better one, but it chooses B. And in fact, the definition of exploration is not choosing the known best action. So each action has some value associated with it. We are expected to choose the best one. But point is, choosing always the best one is not exploration. It's called exploitation. Exploitation, you should choose something that you don't know, that therefore your default value is very low. So it chooses the second best one in this case quite often, but for lesser values of dopamine. And when you go to even lower values of dopamine, there comes when you come to a certain range of very low dopamine, it doesn't choose anything at all, no action at all. So this no action is sort of like your inability to move, and correct action is like you're moving, where you move and make some action. In between, you do something which is not optimal, and that's like exploration. So and that has it looks like a bell curve. We call it the exploratory hill. This exploratory hill is something that emerges in our model. It doesn't exist in any of the previous models, which talk in terms of purely binary terms. A direct pathway is go and indirect pathway is go. In such a picture, there's no question of exploration. That people don't even talk about it. Exploration simply means adding some noise in the output, which comes out of magic, right out of nowhere. So why does this happen? Where is exploration hill coming from? It's coming from the complex dynamics of STNGP. So for high dopamine levels, the direct pathway is selected, and then there's no indirect pathway is kind of bypassed. So you make the better action. For very low dopamine levels, both pathways, the output is not strong enough. In that in the GPI neurons, there's a threshold, right? Only when threshold is crossed, GPI neurons choose an action. So that threshold is not crossed when dopamine levels are low. But for intermediate levels of dopamine, the threshold is crossed, but the contribution is coming mainly from the indirect pathway, which has chaotic or complex dynamics. And because of that, the action selection is somewhat random. So that is what you see in that exploratory hill. So exploration emerges, according to our theory, exploration emerges out of the complex dynamics of STNGP. This complex dynamics is lost in case of Parkinson's patient brains, because you just have synchronized oscillation. There's no rich noise coming out of the STNGP. Therefore, the action selection is not optimal. And if you train the subsequent systems like the cortical systems with that kind of a wrong outputs coming out of basal ganglia, representation in cortex also will be affected, and so that you see a kind of a slow degradation of symptoms, right, in the Parkinson's patient. So this is kind of the picture that we have developed. So with this, we say that the dopamine function will be described in terms of go, explore, and know. So then all this was worked out in the context of like, where there is no learning in what I described. We just have two actions, right? And then what is the effect of dopamine on the action selection? But you need to have a learning model. So this is what we have done here. We applied this to what is called an N choice and N action selection problem, which is called N-armed bandit in reinforcement learning. We did that and we showed how you can train the corticostriatal connections here using this error signal coming from the SNC and so on. So again, we have shown that this basal ganglia model behaves very much like the standard RL models of the N-armed bandit problem. Again, you have two options. You can have epsilon grady policy or softmax policy. So the epsilon grady policy, you randomly choose some non-optimal action with a probability of epsilon, which is a very small value, right? And in softmax policy, you actually have a probabilistic action selection model. So the basal ganglia model that I described behaves just like that. Only thing is the corresponding parameter in the model happens to be the lateral connection strength within STM, right? If you play with that, the system behaves like what you see in the standard RL models when you vary any of these two parameters, epsilon or the inverse temperature parameter. Next, we extend these ideas to continuous domain because N-armed bandit is is a discrete action domain. So whereas in a continuous domain, like which is what happens when you are walking in case of gait or reaching, etc., etc. So we have extended the same theory to continuous domain, and so the equations now almost start looking like simulated annealing, which is like a stochastic gradient descent, right? Or in this case, so what the system is doing. In the previous case, system is picking the action, the discrete action which has highest value. Here, the system is searching for the maximum in the value landscape. Value landscape has a bunch of hills, right? It is searching for the nearest hilltop. Okay, by performing gradient ascent. Only thing is, this is stochastic gradient ascent because of the noise coming from the exchange. So, with this, then more recently we extended this to sequential inputs because the previous inputs are simply static inputs have an input and make an action selection. Whereas a lot of inputs are sequential. So for that, you need to have some kind of a memory sitting in the basal ganglia. So we have invoked the neurobiological fact that stratum has this so MSNs. Some of the MSNs have this property of up-down states where the voltage can be tonically higher. There's a baseline voltage, right, which is tonically high or tonically low for extended durations, and a spiking activity occurs on top of it. It's overlaid on top of it. So we proposed a neuron model called a flip-flop neuron, which can show this kind of a working memory property. And people have also kind of attributed working memory functions to these MSNs, which have up-down states. So with the association, we inserted flip-flop neurons in striatum, and this network can reproduce a lot of very difficult sequential processing tasks. And we also shown how this can explain a lot of working memory deficits in Parkinson's disease in a recent paper. So with that, over the last 1.5 decades, we have explained a wide variety of functions, not only motor, but also several cognitive functions of basal ganglia. We haven't touched affective functions because it's a whole different ballgame, and I don't have much of an idea, and nothing at all in autonomic function. So all this work was summarized in a research monograph, which was published a few years ago. So now for the Indo-Italy project, we have developed a reaching model. Actually, we can extend a previous reaching model to here. So in this case, in the reaching model, we have an SNGP network, which is made up of spiking neurons. These are like Izikevich neurons, which are calibrated specifically for STM and GPE. And so you can operate it in two modes. In one mode, you have regular firing or continuous firing. Right. In other mode, it shows neurons going to bursting, which is the kind of activity you see under Parkinson's conditions. So the bottom one is like normal and upper one is like Parkinson's. So so these are the parameters which you have to adjust in the SNGP part. to produce the Parkinsonian conditions. And also within PD you can have like rigidity dominant case or a rigidity dominant type and a tremor dominant type. So in the model we found that these are the parameters you have to use to produce tremor, right? And these are the parameters you have to use to produce rigidity. And this work is done by Sandeep and others in the lab. So like I just said, you have this continuous firing under normal conditions and this synchronized firing under PD conditions. If you look at the spectrum also, you will find synchronized firing. This is strong energy close to beta, 13 to 20 hertz kind of a band under low dopamine conditions. High dopamine, then that those synchronized firing patterns disappear. And in the case of tremor, you also see this sharp peak around 5 hertz, right, which matches the tremor frequency, right? And let us see what happens to the hand, which is moving towards some target. This is the output of the model in the healthy case, right? And under rigidity case, it's hardly able to move. The joints are rigid. And in the tremor case, you see that small tremor in the hand. So if you measure that, right, it comes to this spectrum, like about 5 hertz. Okay, so that's all I have to share today. I'll stop here and I can take some questions.
I don't see any questions as in the thing, but I'm going to ask you one, if I may.
Please.
You did an arm reaching movement study that basically says BG has a significant role, and then you use rigidity and conditions to model it.
Yeah.
How do you think it'll affect the other loops in the network? Extended interconnected circuits, not really basal ganglia alone.
No, are you talking about the different loops within basal ganglia, or are you talking about basal ganglia versus cerebellum kind of?
Cortex, cerebellum.
So we had because as I understand, like our project is about combining basal ganglia, cerebellum, and spinal cord, which is quite challenging. So we just had a preliminary discussion in the lab recently. So the thing is, my understanding of cerebellum is very, very elementary. It goes something like this. So I guess cerebellum is necessary for ballistic movement. So if you are using, okay. So when it comes to motor learning, right, there are different schools of thought. Our understanding is like this. Basal ganglia is necessary to figure out what is the optimal action. And that happens by this kind of a very slow process of reinforcement learning. You have to try out various actions, figure out which is the best thing, and then discover that optimal action. But once you discover it, you're ready to pass it on to a fast system, which will just kind of mug it up, which is a habitual system, right? This is where the knowledge gained by the cortico-basal ganglia system is passed on to a different loop. What is that different loop? So we used to say that it is a cortical loop, but we never talked about cerebellum before. So another group called by Greg Ashby and others, they also talk in terms of transfer from basal ganglia system to cortical system. But probably it is a corticocerebellar system to which it is transferred, right? So I think we need to, work on those lines and maybe join hands with you and develop that other loop. Another thing is, the cortical loop has long delays, right? That will slow down the system. So cerebellum is supposed to be compensating for those delays by acting like some kind of a Smith predictor. I don't know how far that theory is correct as of now. It's a pretty old idea, right? So if we can incorporate all that and attach this kind of cerebellar loop model to our model, I think that would be great.
Thank you. We should do that. Yeah, definitely. I get your point. And I'll thank you because we have the other speaker also ready.